think we'll be both comparing different sequencing libraries. We chatted a lot about how to profile genomes. We chatted a lot about how to even, let's say, compare samples in CFS session. But what we haven't really talked much about is how to get like different libraries compared and what this can be used for. So today I will start with a short talk where I will first sort of explain of like why we would even care about something like that. And I will then show you finally an example of mixed tissue libraries. With these mixed tissue libraries, it's something that we always wanted to have as a part of the workshop, but we never knew where to fit it so it feels smooth. So in the end, it's sort of like inserted at today's start. Then we will finally look at some 2D Kamil spectra. So Kamil spectra, that is not just a histogram, but these are like sort of one library versus the other, and how can we read them. And then finally, we will jump on practicals, where this will not be really practical in sense of like just tutorials with training, but this will be real data analysis. So you will have to use a lot of things that you already used before. So I hope that you till now you were successful in executing all the commands, because some of them will be useful again. For instance, we will have 8 sequencing libraries, and we would like to we would like to QC them as well. So for example, like I was trying to make very clear from the very beginning that I don't think that there should be any library that won't ever get plotted as a Kamil spectra. So I would like to start with that. But anyway, so now I'm getting ahead of myself. So practically all life, I guess all animal life, is showing this variety of sexual reproduction modes. It's actually really incredible how much variety there is. We have male heterogamy, where the males are defined with XO or XY and sex determination systems. We have female heterogamy, where males have the classically fully diploid karyotypes, and the females are either missing one chromosome or have a W. Or we have even some sort of homomorphic sex chromosomes or environmental sex determination. We have haplodiploids, paternal genome elimination, asexuality. There's just such a huge, enormous amount of interesting sexual reproduction systems. And one thing we really don't understand is why. Because as far as we can tell, sexual reproduction system is the one single most important thing of that like is assumed by all the population genetics models. So how comes that something that's affecting so many things has then so much diversity? How comes that none of these systems is the one optimal system? And so lots of people are interested in studying these sex chromosomes, but to do that you need to sort of like fish them out. So the way we try to do that is by comparing karyotypes. So what I mean by heterogametic sex is that the karyotype of the one sex has one chromosome or multiple chromosomes that are not the same as each other. So while this is the example of Californian stick insects, they don't have 4 chromosomes, but 12. Okay, I don't remember how many chromosomes have Californian stick insects. I'm sorry. But the females are fully diploid, and the males are just missing one of the larger chromosomes, so that it's like XO sex determination. So the property we would be using to fish this XO chromosome out is that the sex chromosome to autosome ratio in males is 1:2. So in theory, that you wouldn't even properly need the female. Even if you would just look at the sequencing library of a male, you see if the sequence is there in a single copy or in 2 copies, because the coverage depth of the sex chromosome will be half over the coverage depth of autosomes. And indeed, this is how it looks like. So this is not a Californian stick insect, because I didn't have any males sequenced. So this is a globular springtail. And I guess I will now try to look as many as you as possible and ask you. So do you think that the genome size of globular springtails, looking at this Kamil spectra that comes from a male, is 350 megabases? So who thinks that it's bigger than that? Put yes. And who thinks this is smaller than this would know.
What's the question?
If the genome size, so this is the genome scope model fitted in a heterogametic sex KMS spectra. And we have an estimate of a genome size. And I'm asking, do you think that this genome size estimate is it's a overestimation or underestimation, or is the correct one? I actually don't know what instructions I gave you on which button you should use, so I will just reveal the answer. Oh my god, I'm so unsuccessful with interacting with you. I'm so sorry. I need to figure out something till next time. So the thing is,
Kamil, you actually have a question in the chat, and I think it might be a good time to address it. So, like, I think lots of people are having issues kind of translating these genome scope curves into how they should think about genome size. Like, my understanding is it's basically like you integrate under the curve, right? It's like the area under the curve is the genome size.
well, the area under the curve divided by the haploid coverage and ploidy.
Right.
I mean, also it's also on the image. You don't have to calculate it. It says length 348,000.
Sure, but people want to understand how this KMS stack is kind of being translated. So I think the easiest way of thinking about it is that your model kind of delimits which KMS are real KMS, and you're basically summing the area under the curve of your model, and that's your genome size, right?
And dividing by haploid and the coverage times ploidy, which is the really important bit there.
And
right. I hope that clarified.
But you also need to multiply by the values on the x-axis, right? It's not just the area under the curve.
Yeah, sure.
You have to multiply.
Sure. It's coverage times number of KMS of that coverage. Yeah, it's not just area under the curve. Yeah, sorry about that. That was not actually accurate at all.
Yeah, and maybe you could just mention something about
that. I will. Okay, so the reason why I wanted to show you is that this picture is because when we were estimating the genome scope models, I was always emphasizing the need of having all the chromosomes in the same ploidy. And in this case, we have a male that has one chromosome once and the rest of the chromosomes multiple times. Actually, it's two chromosomes that are there, just one. And exactly, and the heterozygous estimate is a good question. I will get there later. And basically what the model assumes is that this is a heterozygous peak, although it's not heterozygous peak. It's a peak that is associated with the sex chromosomes. And these are the autosomal peaks. No, this is the autosomal peak. So basically, this is the reason why the genome scope very much underestimates the real genome size, because he thinks that those X chromosomes are simply heterozygous positions. They don't think that they are just homozygous haploid chromosomes. And the same is with the heterozygosity. There is like 1% of heterozygosity. And the reason why it's so high is because it's just assuming that all those haploid gamers that are coming from the sex chromosomes are actually heterozygous gamers, but they're not. So obviously, both the genome size and the heterozygosity estimates are completely pointless when we're looking at this gamer spectrum. But it's still useful. It's useful to see the error rate. It's useful to see there are clear peaks. And yes, errors are not counted in the genome size estimates. So it's basically just a black curve.
So sorry, Kamil, just to make sure, is this what you're expected to see whenever you have sex chromosomes, or
so it depends on like what kind of sex chromosomes you have. So if you have, well, I guess every time you have a haploid sex chromosome, you expect to have like this chromosome to occur in the haploid peak, or like monoploid peak. That's maybe something I should also finally explain. I'm trying to avoid saying haploid peak, because haploid means being half for gamete, while monoploid means being in a single copy. So although I sometimes say haploid, I always mean monoploid. I mean in a single copy. I am glad that somebody is complaining about the fit of the model. Yes, it is a really bad fit, but it's because of a different biological property of this system I will get into in a second. It's because it's a mixed library to give away. But basically what I wanted to say is that the real genome size estimate, or like the real genome size of this species group, would be about 385 megs. So it's about 25 megs underneath, or 35 megs underneath of what we should have found, which is about the size of one chromosome, by the way. And the heterozygous state of the species is about 0.3, 0.35. And this is again like super overestimated, just because everything that is on the X chromosome is counted as heterozygous. So now, this is how a male looks like. This is how a female chimera structure looks like. So you see that this is a lot more. So here the model fits a lot better than in male first, and you can also all those like monoploid chimeras here are really indeed representing the genome, the heterozygous loci. So both the genome size and the heterozygous states were estimated. I have about 10 more individuals that are female sequenced, and all of them are converging around the same values. It's always 0.3 something. So let's get back to males. So there is one weird thing on this chimera spectra, right? Yes. This is how do they look like. These are globular springtails. I spent days and days just staring at those. Yeah, it's a good job. So to explain of why the fit is bad, of like why I had such a weird spacing of the two peaks, I need to somehow think about how the development of globular springtails looks like. All the sperms are fully monoploid. All the eggs are fully monoploid as well. Then when the sperm fertilizes the egg, the zygote is fully diploid. And then females develop normally, but if something triggers a male development, and we don't know what the trigger actually is, the two X chromosomes are ejected in very early embryogenesis. It's like 7 cleavage or something like this. So that you have like just a few tens of cells, and all of them will just eject two X chromosomes. So the karyotypes will be diploid for 4 autosomes and haploid for 2 X chromosomes. All right. And then the second weird thing about globular springtails is that in males, they have in the soma this karyotype with diploid for autosomes, haploid for X chromosomes, but during spermatogenesis, the 2 X chromosomes for some reason co-segregate, which is creating half of the sperm that are fully monoploid and half of the sperm that is missing the 2 X chromosomes. But this sperm is completely inevitable. No, inevitable is that it can be avoided. Inviable, and it immediately degenerates. So and they actually don't even mature as a primary spermatocyte. So only the fully monoploid primary spermatocytes proceed in the secondary secondary spermatocytes and in the mature sperm. So that means that all the sperm that are functional and are still present in the male have this fully monoploid karyotype. It has 4 copies of autosomes and both copies of X chromosomes. And what we think is happening in the species is that they have paternal genome elimination, which is the system where the only paternal genomes are eliminated throughout the development of males. This is found in about 7 different taxa, and the reason why it's proposed in springtails is because in other species where it's really proven, the cytogenetics looks nearly the same during the early development and spermatogenesis. So we think that the first 2 ejected X chromosomes are male, and we also think that the 4 ejected autosomes during spermatogenesis are also male, like the paternal chromosomes, paternal origin. So, which would also mean that the sperm is not just fully monoploid, but it's also all the sperm have exactly the same genotype. They always have only what they inherited from their mom. While in the regular sexual males, the sperm is a sort of a mixture, because every single sperm has a tiny bit of different genotype, which is just a linear combination of genotypes of both parents, but not in these cases. So males contain some fraction of sperm, a sperm that is fully monoploid tissue, which means that the sperm, that if we sequence a whole male, we will sequence 2 different tissues: the soma that has 1 to 2 sex chromosome to autosome ratio, and the sperm that has 1-to-1 sex chromosome to autosome ratio. So we do actually expect that the X to A ratio would be more than 1-to-2. And how much it would be the case will be just dependent on a fraction of the sperm in the body. If the body will be fully just sperm, we will just see 1 peak. If the body will be just soma, we will see 2 evenly spaced peaks. And everything in between just suggests some sort of admixture. And indeed, so this is just replotted the coverage. Now is when I map back reads to assembly, but the ratio between the coverages is still holding. And we see that the autosomal peak is about 30x, and the X peak is about 18.5x. So it's not half, it's not 15x, it's 18.5. So if we would sort of calculate the expected coverages of the individual haplotypes, we expect that the maternal X chromosomes will have 18.5x, maternal autosomes will have 18.5x as well, and the paternal will have about 11.1, which is sort of so then we expect that about the 40% of springtail body are actually sperm to give us this kind of ratios between coverages, which is, I haven't shown the equation, but it's not that, oh, I have millions of questions. Yes, there is no oh, okay. I don't know which one is the first. It is a better, okay. So is this spectra for all genome reads or sex chromosomes only? It's all always for all genome reads. Oh yeah, all genome reads, I believe. It's wild how Kamil spectra can only vary that much, but the interpretations are vast depending on the biological system. I think this is a really huge point of this workshop too. You cannot ignore the rest of the biology of your system. You always need to know what you're doing. And Kamil spectra is just another indication, another sanity check, another way how to look at it. So there is no recombination. Yes, we don't expect any recombination to happen. And that's also the case for lots of systems, actually, that males do not recombine. Off topic to Kamers. Okay, I will skip that one. I guess, but oh wait, off topic. Is there a crossover of maternal paternal X chromosome at any point? Yeah, there should be, but only in females. Sorry, I just thought of this. I guess the paternal X chromosomes which are going to be lost are just maternal ones from one generation ago. That's exactly the case, because the sperm will contain the maternally inherited chromosomes and can generate both males and females, which means that if it's passed to males, then it's lost. If it's passed to females, it's recombined and goes on. Yeah, it's true. It's true. Like if we sequence heterogametic sex, we have no idea how much the first peak is due to heterogametic sex and how much the first peak is due to heterozygosity. That's a very good point too. All right, so I will get back here. So we somehow estimated that there is actually a really large fraction of sperm in the springtail body, and we also have kind of proven this, or like demonstrated that it's probably the case that the maternal alleles have higher coverage than the paternal alleles by looking at the autosomal heterozygous variants. We see that the alleles with the higher coverage among autosomes have about the same coverage distribution as the X chromosomes. So these are the black and red distributions that are nearly fully overlapping. And it's quite approaching our expectations from the calculated ratio of the admixture, which is that dashed line. And the paternal autosomes, or what we think are paternal autosomes, are then the 10xp. So we have not fully proven that which one is maternal and which one is paternal. But at this point, there is no other biological system that would in any way reassemble it other way around. So we kind of confirm at least that all the sperms have exactly the same genotype. So this is sort of like just a biological story about like how we actually used Kamil spectra to discover that something wrong was weird with the library. That we discovered that it all started by just seeing this plot, and I was telling my boss that I don't think that this is normal, that I've seen so many k-mesh spectra, and I've never seen one end peak being shifted towards the diploid peak so much. Like because we have so many of those k-mesh, the means are always very precisely estimated. So it's hardly ever that we see unevenly spaced peaks. So this is the first and the strongest, most evidence for having a library that is of mixed tissues of different karyotypes, that the peaks are unevenly spaced. It was the same case when Lucia was showing you when she had two different plants in there. It's again, she mixed two different tissues with different karyotypes. And what she ended up with are unevenly spaced peaks. I will have one more example later on, but this is sort of just to demonstrate of how it can be shown. And now we will get to how to actually detect the heterogametic sex in our assemblies, or in the reads. So the most common approaches are based on mapping to the reference. So you just assemble the genome, you map the male reads and female reads, and purple is the X chromosome. You expect that the male reads will have half-on coverage over the autosomes. So if you then do male to female coverage ratio, then you expect X chromosomes to be about 1 and X chromosomes, autosomes to be around 1 and the X chromosomes around 0.5. Some people put it on log scale. I actually usually do. So then it's on a 0 and -1. Well, depends on which log you use, but anyway. But the problem with this approach is that if you have fragmented assembly, it can look more like this. So you do see the two peaks. You know for sure that this is a male. You see that at least some of those scaffolds are reliably assigned, probably, but it's very hard to make an exact cut. Which of the scaffolds are actually X-linked and which are autosomal, and then we end up with like lowering the required thresholds of confidence and so I have not estimated male heterozygosity. I suppose that the heterozygosity is a property of a population, so I have only female-based estimates. Sorry.
Thank you.
So here I wanted to say that this method, although it is certainly powerful, and I would not discourage people to use it, I don't think on its own it's giving us enough power to figure it out everywhere. And so the disadvantages is that it's also confusing copy number variations and like the types of structural variations with sex chromosomes. If you will have a male that has a deletion on autosomes, it will just pop up as X-linked if you have just one male and one female. And it does require a reasonably sized scaffolds, which seems that it will be case for most of the bigger animals already, but we're still really far away from assembling all the life. So we will have to, for a while, get used to methods that are a bit more noisy. So the other approach is to directly compare the sequencing libraries. So this is an example, again, of the globular springtail. So this is exactly the two k-mesh spectra you have seen of a male and a female. So this is the one that has the weirdly shifted peaks. And you can see that even here, this is the reference male. So I have like 85x of autosomes and like 50x of X chromosomes, which is again the unevenness I was talking about. And on so that's my x-axis. I have a male here, and then I have a female on the y-axis. You see that the female has just one distribution. That if there wouldn't be no male, I would not see a separation of the two peaks. And this is just simply done by a single command called cat-comp. So you already use cat-comp for comparing genome assemblies and sequencing libraries. So this is in general just comparing basically two sets of sequences, and you can give it whatever you want. And with different parameters, it generates you different plots, but you don't even have to give it the parameter. And if you just do cat-comp and two sets of reads or two sets of sequences, it will just generate you the 2D Kramer matrix which is like just a matrix gives you basically the values for the individual tiles on this plot. And then you can just plot it with other subcommands of cat, because they have multiple different visualization tools for the same. But so generating the 2D K-mesh spectra is in fact quite easy. The reading it is actually not that difficult either. So as I said, the bottom one is male, the top one is female. And here I annotated it for you. So oh no, this is A-link. Does everybody see yeah, okay. So that's something that should not be missed. So okay, so here I have all those autosomal K-mesh, those that are present in female and in presumably diploid state, and in male also in diploid state. There will be a bunch of K-mesh here, and here there will be just heterozygotes in female and X-linked in male, by the way, just a full disclaimer. But most of these K-mesh will be the X-linked, because they will be those that are in male haploid and in female diploid. Then I have this red square over here, which will be mostly the sequencing errors. So the K-mesh that are in low coverage in both males and females. And then I will have lots of these K-mesh, well, lots, it depends, that will be specific to individual libraries, which will be, in this case, only the genetic differences between the two individuals. And but in some other cases, for example, if I would have XY sex determination system, I would also have here, so the male specific would be also the Y chromosome. So it would be a combination of population variability and Y chromosomes. But again, you need to know what samples you have sequenced. So I know that the globular spindles are incredibly hard to rear in the lab. So I have wild caught population. How this would look like, female versus female or male versus male. Well, it would be a tiny bit more boring. You would have one big peak for all the diploid stuff for two females, and then you would have a bunch of very subtle K-mesh. So it would be just very subtle clouds of those that are heterozygous, or heterozygous in both. Probably that wouldn't be even seen there at all. And again, the genetic differences. So you would just not see the X-linked practically. And a male versus male, that's actually a good question. Yeah, I think you would just see both of them being haploid and both of those being diploid. So I suppose that the X-linked cloud would just shift halfway down. But again, we can actually try it out. Once we know which of the tsetseflies are males and females, we can generate any combination. All right, so now let's get to it. Like I bet everybody's dying to do their first 2D K-mesh spectra. So because this data is super exciting and new, they are confidential. So I guess that's the price we have to pay for novelty. So I just ask you not to download it. I think you can still show or your colleagues the plots. That's fine, but just the data should stay protected. And I would get into trouble if they would get out. So please don't. I would also like to describe that I really don't know the data. I haven't analyzed them at all. It's really on us in these next 2 hours, and we have so let's divide in 8 individual breakout rooms, because we have 8 individual sequenced, and let's analyze them. Let's divide them into 8 breakout rooms, and let's hope that we will get everything done quite soon. Divide and conquer. Can I see some thumbs up? That sounds good. Excellent. So your task will be to first do the classical QC, like 1D K-mesh spectra, then we should guess the sex of the sample. Perhaps from the 1D chemistry structure, it won't be obvious because we won't be able to tell whether we are looking at the heterozygosity or the X chromosome. So we can do 2D chemistry spectra, and that will probably reveal us both. From the 2D chemistry spectra, it should be clear which one is which. And I was told which sample is which sex. That's one thing I know, but I think we should be able to get it from the data, so I didn't tell it to you. So then we will collect the data in a table together. And the tutorial today is very verbose. It was the first tutorial I wrote. So it's sort of explaining everything on its own. You don't have to really pay that much attention on to the smaller text. I try to highlight the important bits in Testify. But then what we would like to do afterwards is to define coverage ranges of which we would like to extract the specific k-mers that are like the chromosome specific k-mers. So, and just for the record, this is not just about the sex chromosomes, because there are lots of other exciting chromosomes like B chromosomes that are polymorphic between different lineages. So when basically the structural variant in the population is actually the presence or absence of a whole chromosome. Or even if you would have like a very large inversions that could be detected in a similar way too. I have seen it done on species that had like two variants of females with a huge inversion, and it worked out too. And as you have seen the talk of Christina, it can be also done on a germ-line restricted DNA. If you compare the reproductive tissue of the species with some sort of somatic tissue, which was had in the case of fungus and Christina. And yeah, I guess you could take any part of bird, you don't have to decapitate them. So the example Christina gave you was the germ-line specific chromosomes of Sierra fly. And here is a schema of what kind of karyotypes are expected to be in these two tissues. In soma, you have 1:2 ratio of X chromosomes to autosomes, and in the germ, you have 2:2:1 germline to X to autosome ratio. So if we look at the chemospectra of the soma only, what we see is evenly spaced peaks, one representing the X chromosome, one representing the autosome. So far, so good. If you look at the germ, however, we don't see them spaced 1:2. Although like there is nothing else than things that are in 1 copy or 2 copy, we actually do see them sort of like oddly spaced from each other. And the reason is because the germ library is not really fully the germ library. If you tear out the testes, you get a lot of other cells that are somatic as well. And the thing is, it's actually more the soma we sequence than the germ-line. And wait, how can I know? Well, because I see that the autosomal peak is actually larger, like it has a higher coverage than the X peak, although I know that in a germ-line, the X chromosomes are there in twice higher count than the autosomes. But you also see that the two peaks are very much blended together, which means that it's not, again, 1:2. It's like 1:0.85 or something like this, a ratio between the autosomes and the X chromosomes. And then here you have the peak that is representing the GRC chromosomes. So there are two different reasons why it's so much shifted to left. And one is the one that Christina already explained in her talk, and it is that the two GRC chromosomes are actually two different chromosomes. So this is actually not a diploid coverage of GRCs, but haploid coverage of GRCs. And actually that's it. But you kind of again see that the indication that this is a mixed tissue library comes from the fact that the peaks are unevenly spaced. And perhaps if you were wondering of how do we know that the right one is autosomes and the left one is X chromosomes? Well, we know it from the 2D frequency spectra, because on the Y axis we have the germ, on X axis we have the soma, and in the soma we know that the right peak is autosomal for sure and the left peak is X chromosome for sure, because that's just the karyotype is clear in the head. There is no chance of any type of contamination unless we would have some sort of like external to fungusness contamination. And so we know that these are the autosomes and these are the X chromosomes. So you can clearly see that the coverage even in the germline of autosomes is higher than of X chromosomes. And then we have the GR6. So now I would question: could you remind us again the reasoning behind why lower peak is haploid coverage? Well, because that's okay. For this, I will actually close this presentation and show you the previous presentation because that was, I think, better explained. Give me a second. That was the introduction. Yeah, as you might have seen, I really do like Google Slides. So this is the main reason why I showed the, I guess I can copy this slide to the other presentation. Okay, never mind. So every time we take a piece of tissue, every single cell has the number of chromosomes like this, like in each cell. So what would happen if of the smaller chromosome, I would have just one copy in each? Then if I would fragment the DNA and just get the reads out of the copy number, I would be twice more likely to take the bigger chromosomes than the smaller chromosome, which is the reason why I have twice higher coverage. Well, the coverage is always dependent on how many copies of the DNA there are in each cell. Does it make sense? That's actually absolutely essential point of this comparison of multiple libraries of different karyotypes. Does it make sense? Can I have a yes, okay, perfect. It did. It's just like this is such a fundament that I really needed to make sure that we're all on the same boat, I guess. We're all on board. I'm so tired. I'm so sorry. So fair enough. So the main reason why I wanted to re-explain this fungus nuts and show you the example is just to give you the idea again how to detect mixed tissues and how to sort of deal with it. The fact that it's shifted, it actually doesn't have to be a big problem if you know that it might be a mixture, and if you know that the two karyotypes can possibly be there. But you can also use it for discovery, but then it's a tiny bit more complicated. And again, I don't want to advise you against using the mapping approaches, but I do think the biggest power is even using the two approaches in combination. So the pitfalls of, so actually what I haven't explained is that what we are going to do now is to define the coverage ranges of the individual gamers that we think they will be X-linked or not. And if you, although in this case we had just two libraries, so we sort of defined it just for the two, we can define it for as many libraries as we want. And we have 8 libraries. So for every single sample, we would like to define the left boundary of what's the minimal haploid coverage, what's the minimal maximal haploid coverage, what's the minimal diploid coverage, and what's the maximal diploid coverage. So then we will count for every single KMA how many types is in male and female reads. And then what we can do is just then to say, give me all the KMAs that are found in one genomic copy in at least 3 out of 4 males and 3 out of 4 females, or something like this. So we can just use the power that we have more than one individual just to sort of like decide of which KMAs are really sex specific and like get the purity out of the number. The one problem you can complain about is the sort of the arbitrarity of the process. And as far as I know, nobody actually figured out how to do this well probabilistically, or like having a model that would explicitly model it. And this is actually something that I would like to do in future. So don't try to figure it out on your own. If you would like to do, please contact me. I would feel left behind if you would figure it out on your own. And also, one small aspect I wanted to point out is that when we were looking at the springtails compared with each other, we had these lines of genetic variability between the species. Would you guess why there is none over here? Like why there is no camas that are in female, no, that are in heads, that are not in germline? I guess nobody wants to answer, but it's just because these are lab lines. They are genetically completely uniform. There's no population variability, so obviously there are no other camers in the germline. If there would be population variability, it would be a tiny bit problematic because the GRCs we would have in this distribution would be mixed with the genetic variability of this individual, just simply the genetic differences that are between them. But luckily, in this setting, it was not a problem at all. So the pitfalls of this are if the divergence of chromosomes is too small. So if the chromosomes we're trying to isolate has two similar sequences to the other chromosomes, then instead of having them isolated as a single copy, we will see them shifted somewhere here by like by a coverage of the autosomes or X chromosomes and the shared sequence. If you would be wondering about how to isolate those, I would recommend to talk to Paco, who's over here as well. He's done this in the zebra finches. And again, the pitfall that you can face with any kind of camera approach is that if your coverage is too small to separate the individual camera clouds or peaks, then you don't have a sort of a reliable sets of cameras you could start with. And for the rest of the day, what we were trying to do is to match these isolated cameras. So once we isolate the cameras from the dump files you have hopefully created, we will match them to the genome. And for this, we use a quick and dirty approach. It was I asked this question a while ago, and Daniel gave me a great answer. You can just tweak BWA-MEM to map cameras, like not to map cameras, to match camer to the assembly. And basically the hack you're using is that you're just exploiting the fact that you can set up the size of the seed. So basically we're saying that every single read, which is not read but a camer, will be mapping, will start the alignment anywhere in the genome only if it will match by 21 consecutive bases. So if I would have if I wouldn't have 21 MERS but 27 MERS, I would have to set it up here, right? But I will just set this number to the same length as are my as is the length of my camer, and then I said T21, which is like the minimal required confidence in mapping, which is the same as the length. So it's just like, it just literally finds all the matches, which is just very handy because BWMM is amazingly fast. So then this will like just shoot the camers back on your genome. And basically this is where I run out of slides because I don't have anything to show you before on some camer plots. All right, can people talk to me? Like literally unmute themselves and say once per while something. I need to hear from you. What do you think that so we have a bunch of camer spectra, and you see that some of them have the left peak quite high, some of them the left peak quite low. So could you tell me what do you think is the sex of the individual samples?
Is this a male?
Yes, I do think that will be a male. Like the heterozygosity up to 2% is really high. Like it's not impossible, but if we look at the other sample, the heterozygosity is a little lower. So it's most likely that it's just simply that this peak is partially the X chromosome. And the second indication, so we don't have to rely only on the heterozygosity, is the genome size. You see that this has just 385, and this has 416. 6 genome size. So I suppose that this is a male and this is a female. So this is number 17. This will be another male. This is number 15. Another
female.
Yes. We have a mitochondria error and another female, and we have a 2D spot. Absolutely amazing. So this is 12 versus 14. So now we quite clearly see of which one is male and which one is female. Could somebody tell me?
Male Glossina 12
Yes,
and Glossina 14 is female.
that's right. So now if we take a look on the margins, we see that there is a bit of population variability, but we also see that the males have a lot more specific, the male specific gametes. So this is our Y chromosome. So would somebody advise me on what cuts they would choose to choose the X specific gametes from the individual samples? I guess if we would, so here we have a sample gametes that are in a single copy in both male and female. So those that are most likely heterozygous gametes, and it will be probably a combination of those that are also heterozygous in males and combination of those that are X-linked. The same applies here, but I would just for sanity just take a square like this. Can you see the square I'm drawing? It's just a screenshot cursor. For autosomal gametes, I would take a square like this, I guess. We could.
Oh, no, we cannot see the square actually. Just your
Okay.
person.
So I would. So I guess for the haploid in males, I would take it from 20 to 40. And for diploid in males, I would take 40 to 80. Oh, sorry, 60 to 80, I guess, or 50. Well, I guess if we take 50, we will have there a bunch of X-linked gametes too. If we're happy with that, that's fine. But we will miss a few autosomal. So we just, again, need to make this choice. It's like, it is, again, the biggest downside of all the chemospectral analysis. We need to make the cutoffs. We need to make some arbitrary decisions. Is this analysis only works for high coverage data? Well, what does it mean high coverage? I would argue that 40x is not that high coverage, but it is. It cannot be like skimming data. This is like for no genome skimming. So we have another male, another female. What is that?
I don't know.
I'm sorry, I haven't ever seen anything like this before. It honestly looks like a glitch in the code. But I'm suspecting that the y-axis, that's the 12. We already know that 12 is male, right? And 15 then looks like a female. So why does it work? And I guess that's it. This is really weird. So in this case, I would take the matrix file and find the cat function that replots regenerates the plot and redo the plot again. So
it's the second plot, so it's redone.
It's what?
Redone. I mean, it's with the second command you told me.
So if you do this cat com, it's just something that generates a matrix file, and if you also give it some sort of arguments, it also generates the plots. And the argument we were giving it is n. which generates this kind of plot. But you don't have to do both steps at the same time. You can first generate the matrix files, and I think then it's cat density, but I'm not entirely sure. Like somebody should check in the manual and inputted the matrix file, and that will again regenerate this plot from the matrix file. So I think there was just a glitch, and if you take the matrix file, you don't have to like recompute the 2D matrix. You can just like replot it. Does it make sense?
Could be.
All right. So now, not totally related really, but meta-plastic genomes, you have no effects for. Okay, so the mitochondrial and plastic genomes will usually appear in chemospectra too, but they are usually a lot higher copy number than the genome. So they will be somewhere like far on the right. So usually on the linear plots, you cannot see them. On the log plots, you can see them. And in my experience, they have some effect on the genome size estimates, but usually in an order of max. Like it's not usually something huge. So if you want, you can get rid of the plastids, but for this high complexity part of the genome, it usually does not make any difference. Sample 12 again. So in the best viewed on the 2D plot. Here, we have up to 45, the X-linked monoploid k-mers in males, and up to 80 diploid k-mers in male. Perfect. How do we call it in the, so now we can also do the sample number 14. So let me young the word, Glossina, young word, paste, Control A. That's why I like Vim. Is it male or female? 12. That's female. And here the monoploid coverage would be, so the coverage is a bit lower. You see that we are on like the diploid will be, I guess, 20 to 40.
Yeah.
So we're going to 20 or 40. That's the easy one. And the more
20.
10 to 20. I would probably do 5 to 15 actually. I would just leave there a margin of those that we just don't know. What do you think?
Fair enough.